Amazing. So this is a real hybrid event. Thank you, everyone, and thank you for attending. My name is Micha Breakstone. I'm the CEO and co-founder of NeuraLight. Our mission is to transform the lives of billions of people suffering from neurological disorders by digitizing neurological care. And this talk will be about how we can leverage AI and computer vision, data science to digitize neurological care. The outline of the talk will be, I'll give you some quick context on the challenges for neurological drug development, just so you understand what we're trying to solve. Then delve into the science and talk about the power of oculometrics, namely the power of micro eye measurements. And then I'll pass the microphone over to someone much smarter than me, that's Vova. He's our founding engineer, and he'll be talking about how you can use AI and computer vision to augment oculometrics and actually achieve higher impact and solve the problem at stake. So in terms of context, first of all, just about neurology. So neurological disorders are the leading cause of global disability. Over 1 billion people worldwide and 100 million people in the US suffer from neurological disorders, and the cost is over $800 billion in the US alone. At the same time, neurology is, way back. It's probably 50, 60, 70 years behind most other areas in medicine. Screening, diagnosis, and care are extremely subjective. There are mostly no real objective measurements. They're dependent on manual examination by experts. Multiple signs and symptoms are not easy to elicit or detect. And this creates a huge challenge for drug development, for clinical trials, for precision care for neurology, and for monitoring of treatment. I'm not sure if you guys are following the news, but about 3 or 4 weeks ago, maybe a little more, the FDA approved for the first time in 18 years a drug for Alzheimer. And that's kind of a sign of how hard it is to approve these drugs. It's not for lack of molecules. It's we'll talk about what the reason is in a bit, actually. But lack of subjective and sensitive measurements. So if you look at the current state of neurological drug development, there are 3 main pains. One is that promising clinical trials fail. The second is that clinical studies are extremely expensive, twice or thrice the price of oncological trials, and time to market is also unnecessarily long. And when you ask why, when you ask execs at pharma or neurologists developing at pharma why, the answer it boils down to is the flawed measurement. Basically, the endpoints, namely the measurements that are used by the FDA to approve or to ascertain the acuity of a disease, are extremely suffer from extreme high variability, both inter and inter-rater variability. The proxies are bad predictors for success. There's no robust profiling, or in medical terms, phenotyping. So it's very hard to identify what populations the medicine works on better. High variability also forces large trials with many participants because in order to elicit a statistically significant result, you need many people. The low variability proxies are extremely expensive, like MRI. And because the measures today are subclinical, namely they can't measure anything that you don't see, you actually have to wait for the disease to unfold. And since these diseases usually don't, unfold over days or weeks or months, but years, the trials are extremely long. This is just an example for a specific disease, MS, multiple sclerosis, but what we're saying now is actually true for a very wide range of neurological disorders, everything from Parkinson's, Alzheimer, and other dementias, ALS, Huntington's, MS, of course, and also some diseases that are more borderline with psychiatry, like schizophrenia, and even to some extent depression and anxiety. Walking into the science, so video markers are, and specifically oculometrics, micro measurements of the eye, have been proven beyond doubt. There's a huge body of literature showing the deep correlations between micro movements of the eye and the progression of various diseases. This is again an example for MS. So here there are over 150 papers for multiple sclerosis showing beyond any doubt that there are deep relations between the progression of the disease as measured by EDSS, a measure of disability, and other measurements, and micro movements of the eye. Here, there are 150 papers. There are 6 examples here, including from very high impact factor journals like Nature Reviews and MS Journal, Frontiers. For other diseases, I think there are over 700 papers, including from Lancet Neurology, Brain, Neurology itself, and other top high impact factor journals. So the science is quite robust. The advantages of oculometric markers are also very clear. Basically, they solve everything that is broken. So they're objective with very low variability, both intra and intra-rater variability, so you can actually get robust results. They're sensitive and accurate, so they're able to measure things that are entirely subclinical, allowing for shorter decision times. And just as importantly, they provide a massive layer of data. And this data allows for, what a layman would call profiling, or in medicine we call phenotyping, in order to build more robust trials that have a higher chance of success. And with that saying, I'll move the microphone over to Vova. Vova is our founding engineer and has helped from day -1 basically help us build the system. So thanks, Vova, and thank you guys.
Thank you very much, Micha. I would like to continue by diving a little deeper into the current state of the art and what you're doing at NeuraLight to bridge the gap between the science and wide real-world impact. I will be giving examples from MS. It's only one of the diseases that we work with, but the concepts apply to other diseases as well. We see in the literature above 100 different oculometric measurements, namely a measurement of some movement of the eye, and all are shown to be deeply correlated with EDSS, FSS, PASAT, all of which are scales at which MS patients are measured to identify the progression of their disease, whereas EDSS is the most prominent one. I'm showing you here one representative study out of many, that measures some oculometric parameters, SACADs, and explores the correlation in some sense to EDSS. So we see here 5 columns. Each is a different measurement, different oculometric measurement, and it has 2 subcolumns. On the left, there will be early stage MS patients, and on the right, advanced stage MS patients. And what we clearly can see in common is that the distribution of the measurement is separable between both groups in each of the parameters, even with a naked eye. This gives us a very strong indication that these parameters carry information that is deeply informative of the actual disease progression. The results repeat themselves across multiple independent studies and all the oculometric measurements, which is a very good sign for us as well. Moreover, we have evidence that incorporating multiple parameters at the same time drives accuracy upwards. We are showing here a comparison of 3 different models that run logistic regression on oculometric parameters to predict whether the EDSS of the patient changed between 2 encounters. The blue line here uses just one parameter. Think of it as if just the left column from the previous slide. And that's the ROC for that model. Actually, most of the studies currently use just one parameter for this purpose. But luckily for us, there are some examples that we find in the literature that use more parameters. The green line uses 2 parameters. Just envision the 2 columns in the previous slide. The model is clearly superior to the previous, and so forth when you use 3 parameters. And we have strong reasons to believe that as we continue incorporating more measurements into the same model, we will be pushing the ROC towards the top left corner and improving accuracy. Next slide. So to give you a peek under the hood of how these measurements are performed currently today at lab settings, it will be similar to what you see in this picture. The patient will be requested to perform some task on a monitor. Their head will rest on a chin rest. It will be restrained. There will be an expensive dedicated device to measure the specific measurement they're performing at this study. And unlike in this picture, the room will be usually pitch dark. This is inconvenient, to say the least, for the patient and burdensome, and it is not reproducible at all in real-world settings. So here comes in our technology. First and foremost, I'm happy to introduce you to Eddie. He is NeuraLight's co-founder and CTO. That's Eddie. And maybe after a few shots of whiskey, according to the legend. But what we are demonstrating here is the technology that we developed to extract with high accuracy the econometric measurements using a regular smartphone or web camera. In a nutshell, our technology, we use off-the-shelf machine learning to extract facial landmarks and localize our computation around the eyes area. And then we add an additional layer that utilizes the unique geometry of the eye to further stabilize the measurement and even achieve sub-pixel resolution. Putting these points together, our technology removes the friction and the burden we described previously. It even allows to take these measurements remotely with a device that almost everyone has. Moreover, since we are not doing this on a dedicated device, okay, I'll end very shortly. Since we are not doing it on a dedicated device, we are able to extract multiple parameters at once. And the last thing that I would like you to keep in mind is this slide, is why we're doing this, why this is important. We are able to give value throughout the drug development cycle with two very important points. First is that it's seamless, and second is that we can do it immediately without the need for explicit FDA approval from early phase 1 and 2 trials to post-market surveillance and patient compliance. So with that, I'll end. And do we have time for questions? Happy to take questions afterwards. Thank you.